Welcome to the World Potato Congress webinar series. My name is Eric Magembe. I'd like to remind you to mark your calendar for next year's World Potato Congress that is going to happen in Australia from June 23rd to 26th. Next slide, please. Thank you again for attending the webinar series. If you have any questions, please submit them to the Q&A tab and we'll be able to address them at the end of the presentation. This presentation is going to be recorded and you'll access it through the website. We'd like to acknowledge the various sponsors and partners that are supporting us. And we can't name all of them, but you can see the slide there. I'm going to do this presentation together with Marc Ghislain. We are from the International Potato Center based in Nairobi, Kenya. And we're going to talk about the deployment of lead blight resistant potato, biotech potato in Africa. We are on the Feed the Future Global Biotech Potato Partnership Project that started in 2021, going through 2026. We are being supported by USAID in this project, and we are various partners in this project, being led by Michigan State University. And the PI is Dr. David Dauches, who is the director of the project, and we have 2 deputy directors and several global resource leads in pathology, in molecular and technical support, communication, project management, monitoring and evaluation, and also financial management. So lead blight is an old disease. It has been there for a while. It is the disease that caused the famous Irish potato famine that happened in 1845 to 1849, that led to the death of over 1.5 million people at that time. And over 1 million people immigrated to North America due to the famine that was happening. As you can see in the colored picture there, the disease causes a lot of destruction to the potato. And the losses are estimated to be over $10 billion worldwide every year. So the disease is mainly managed through the use of fungicides. So farmers have to apply fungicides. And in the developing world, some farmers do apply the fungicide without following the recommended regime. As you can see in that photo, some farmers start to spray even after the disease has manifested. And in subtropical regions, growers must apply 2 to 3 times a week to keep the disease under control, which translates to about 20 sprays per season. And this is about 40% of the production costs. And for the smallholder farmers, this is a lot. And some of them, they end up not spraying using fungicide, and they can lose their crop. And if the disease manifests without any control, it can lead to the total destruction of the crop in 2 weeks. So lead blight is caused by Phytophthora infestans, which normally infects the potato leaf, and it secretes specific proteins that are called effectors that help it to invade the plant and obtain nutrients from it. The plant, on the other side, can overcome this pathogen by producing R proteins that are from R genes. And these R proteins, they recognize the effectors and lead to effector triggered immunity that eventually leads to the cell death, therefore depriving the pathogen to get nutrients, and therefore it doesn't spread. So over the years, over 50 R genes have been identified, and as we go by, more are being discovered in potato and other wild relatives. So breeding has been utilized in trying to in crop improvement. And in breeding, you can transfer your desired gene by crossing into your commercial variety. However, breeding leads to this genetic drag where you have your desired gene also is transferred along, or it's spread along with the undesired gene. And this genetic drag has to be eliminated by back-crossing your new variety until you are able to get rid of the undesired genes. And this takes a while, it takes a long time. Unlike in plant biotechnology, where the desired gene is identified and is cloned and transferred using various methods to the commercial variety, and the new variety has only the desired gene, and this usually takes a short time as opposed to the traditional plant breeding methods. So this is the approach that we have used. And previously, breeders, next slide please, have tried to breed resistance to lead blight using conventional breeding methods, like the varieties Bionica and Toluca. These varieties were bred from Solanum balcostanum, which has natural resistance to lead blight. And it took 46 years to breed these lines. So it takes a long time by conventional breeding, although this can be accelerated using marker-assisted selection and other newer breeding tools. And we have 50 potato varieties that have been found to bear these resistant genes, either single or multiple genes. But it takes a long time to develop them, and the durability is not usually guaranteed because these are single genes or other minor genes that can easily be overcome by the pathogen. Next slide. So our approach has been to use resistant genes from wild relatives of potato, and we have tagged these genes, 3 genes, from 2 from Solanum balcostanum, that is RB, and RPI BLB2, and also the third one from Solanum venturi, that is RPI BNT1. So we stacked these 3 genes and we transferred them to farmer preferred varieties, that is Victoria Sante, Tangi, Tigoni, Desiree, Jalene, and Diamond. This can be achieved in 2 to 4 years, depending on the lab. If the lab has resources and facilities, it can be achieved in a short time of about 2 years. The target countries for these are Kenya, Rwanda, Uganda, Ethiopia, and Nigeria. So we started to test these biotech potato in Africa, in Uganda initially. And in Uganda, we partnered with the National Agricultural Research Organization from 2015 to 2021, led by Alex Bereje and Abel Arinaitwe. And throughout these years, we did 17 experimental field trials in 3 locations where potato is grown in Uganda. And throughout the experimental trials, we demonstrated that there was single complete resistance to lead blight by these biotech potatoes. We saw good agronomic performance for one lead event, that is lead blight resistant biotech potato, Victoria 172. And you can see from the pictures there that it is completely resistant even under natural infection. And you can see the plots there where we don't have any plant. This has been wiped out by the lead blight, and the green plants that you see there are the Victoria 172, which is completely resistant to the disease throughout the season. Next slide. So we also are doing the work with in Kenya with the Kenya Agricultural and Livestock Research Organization, and we started partnering with them from 2010, doing some laboratory and greenhouse work. And in 2021, with the new project, we started doing field trials with Dr. Catherine Kataracha and Moses Nyongesa. And we have done 4 experimental field trials in 3 locations in Kenya. And we have revealed that there is good agronomic performance for 5 lead biotech potatoes. However, in Kenya, we have not, for the 2 seasons that we have tested the biotech potatoes, we haven't seen lead blight because the conditions were not right for it to grow, but we got very good regulatory data. We have also tested the biotech potatoes in Nigeria. And in Nigeria, we partnered with the National Root Crop Research Institute, led by Dr. Charles Amadi and Kaya Shuaibu, since 2021. And in Nigeria, we only managed to do 1 season, 1 experimental field trial in 1 location, that is Jos Plateau, where potato is the main crop being grown there. And also in Nigeria, we showed complete resistance to lead blight disease in 4 biotech potatoes, that is Diamond, Victoria 185, Shangi 6, Shangi 15, and Tigori 254. And also in Nigeria, we were able to show that the lead biotech potato were completely resistant to lead blight. As you can see there, the green plots are the biotech potatoes, where the non-biotech potatoes, the conventional ones, are wiped out at about 60 days in the field. Over to Marc Ghislain to continue with the rest of the presentation.
Thank you very much, Eric. So in this second section, we will focus on the next steps for the VIC 172, which was identified in both Uganda and Kenya as a lead biotech potato. But first of all, I want to address one critical question, which is the durability of the biotech lead blight resistant potato. It is a question that comes over and over. And I want to simply present 4 reasons why we think the technology will provide durable resistance to lead blight. First of all, the R genes that we are using are from wild relatives that have never been widely grown in cultivated areas. And even though there might be some strains that are virulent against these R genes, none of them have been found in Africa so far. The second reason is the resistance is based on 3 R genes, not simply 1, which makes for Phytophthora infestan much harder to overcome the resistance. The third reason is that we select on purpose completely resistant biotech potato, and we do not favor the selection of tolerance to the disease to minimize the risk of adaptation of the pathogen. Finally, perhaps the most important point is that should we see that Phytophthora infestan is able to overcome the biotech potato, a new biotech potato with different R genes will be deployed to replace the defeated biotech potato. So we think that with for these three reasons, we can have durable resistance to lead blight. So the current step in which we are is to develop studies to demonstrate safety to human and the environment, to develop a regulatory dossier which will be submitted to national competent authority for approval of this VIC 172 for release to the environment, cultivation, and consumption. There are basically 7 studies that I want to present today. First of all, allergenicity and toxicity. We have demonstrated that there is no allergenicity or toxicity known for this R protein. This has been done by bioinformatics search as well as by a weight of evidence. Indeed, the potato is loaded with thousands of R genes that are similar to the one that we have been used. In addition, two of the ones that are in our biotech potato are already in conventional potato varieties that are commercialized and being consumed, Alouette, Toluca, and Bionica. In addition, the mode of action is quite important. Our proteins are not toxins, they don't kill Phytophthora, but they are triggers of plant cell death. So this is quite important and very different from, for example, the Bt toxins that are well known in GM crops. Third very important point is that our proteins are barely detectable. They often are referred to as intractable, and we think they are most often below the threshold for toxicological concern. The second study that we have completed completely for VIC 172 is to characterize the DNA inserted into the potato genome and its stability. We have shown that the DNA is inserted intact. There are no mutation. All 3 genes are fully functional. And we even show that it was inserted in chromosome 6. The precise position has been identified, and that no allele of an existing gene has been interrupted. Over time, we've seen that this insertion is stable, and we observe a low gene expression as expected for this type of technology. Another study that has been completed has been the demonstration of complete resistance to late blight of Victoria 172. This has been done both in the lab, in the greenhouse, using detached leaf assays with Kenyan strains, whole plant assays, as well as in the field now, as Eric mentioned, in 4 countries. So it has been developed, and we have published these data. Just some aerial picture of what we have done. With no fungicide protection, it's a very black and white situation where the Victoria 170 is completely green, unaffected at all by Phytophthora, whereas the conventional variety, if not protected by fungicide, is completely wiped out. These are pictures from different trials in Uganda. So complete resistance has been fully demonstrated for VIC 172. Perhaps the most complicated and time-consuming study is phenotypic characterization and agronomic performance. This is to demonstrate that there are no significant difference in the phenotype and performance of the biotech potato compared to its conventional variety. So basically, we test VIC 172 against Victoria. We did it in 3 locations, 2 seasons, and 3 repetitions. And so we looked at parameters for plant growth, for yield, for phenotype, and recorded them on a weekly basis. Here, just the most important one, perhaps, the yield. As you can see on the picture, when treated with fungicide, we visually don't see any difference between VIC 172 and Victoria in growth. And indeed, when we look at the yield, we didn't see difference between VIC 172 and Victoria. We saw, of course, some difference between locations, but not between the 2 genotypes. So basically, the conclusion of that is that the stack of the 3 R gene doesn't cause any yield penalty, which is quite important also. Then the impact on the environment. When we look at the mode of action, there's actually no really credible, plausible hypothesis of an impact on non-target organisms, because the mode of action is extremely specific to Phytophthora infestans, and not even to all Phytophthora infestans, to specific strains of Phytophthora infestans. So nevertheless, we have to make some observation just to make sure that there is no unanticipated or unexpected impact. And we did this at the plot level for different arthropods, including diseases, and we didn't see so far any differences. So the same weight of evidence for no toxicity applies to impacts on non-target. To look at the issue of weediness or invasiveness, it's always good to look at the crop's reproductive features, such as flowers, berries, volunteers. We didn't do any gene flow studies because it's not relevant to Africa, where there are no wild relatives, sexually compatible wild relative of the potato. And again, on the reproductive feature, we didn't see any differences. And another important study to conduct is to look at the composition analysis of tubers from Vic 172 compared to Victoria. We did that in our nutritional lab in Beka in Nairobi, Kenya, following two OECD recommendation documents, the one in 2002, which recommended us to look at 5 components, and a document, a recent update of this document in 2021, which recommend a bit more, a list of 11 components. So we did observe from time to time some variation between the Vic 172 and Victoria, but first of all were within potato ranges, and none of them were biologically meaningful. Right now, there is a ex-ante socio-economic analysis going on, which is important to assess the impact of the deployment of the biotech potato in the target country. So this research is being conducted right now through literature review, data collection, and validation through potato experts. So we do not have yet numbers to share, but it's clear that this technology will bring yield gain, will reduce production costs, and will have health benefits. So I want to conclude with the 3 major points about this technology. The first one is that this technology has been tested around the world and proven to confer complete and possibly durable resistance to late blight disease. As you see on this map, it's about 10 different countries around the world, in between America, Europe, Africa, and Asia, where this technology has been tested and has provided always the same result, complete resistance to late blight. A second point which is quite critical, is that the studies and the cost associated to the regulatory studies, which are required to deregulate this specific biotech potato resistant to late blight, are within reason. It is not the level of multiple dozens of millions of US dollars that are needed for other crops. And finally, I want to try to make the parallel between the adoption of potato in France in the 18th century, when the ordinary people believe potato was toxic, was responsible of lepra, and was not compatible with their religious belief, because it was not mentioned anywhere in the Bible. Well, thanks to Parmentier and Louis XVI, potato became what it is today in that country. Well, I do believe that the same applies to GM potato, to our biotech potato. It's important that political leadership is being present in the country where we want to deploy this country to have a successful adoption. So I want here to briefly, quickly acknowledge our sponsors and partners. Until 2021, we were funded by USAID, Two Blades, and USDA and the CRP research program on roots, tubers, and banana in a partnership between NARO from Uganda and CIP. And since 2021, we are operating in Africa, in Kenya and Nigeria, under the sponsor of USAID, with the in Kenya, the organization CARO and NRCRI in Nigeria, and we have an important partnership with AATF. All of this, as Eric mentioned at the beginning, under the leadership, the global leadership for Africa and Asia of the Michigan State University. So this is the end of the presentation. I think that now we will switch to your question. Many thanks to your attention. Feel free to add more questions in the Q&A tab. So maybe I will ask Eric to look at the first question.
Thank you, Marc. There are no questions so far on the Q&A. Please feel to, okay. Okay, there's one question that has come up. How can you move the 3R week ahead in Uganda?
So maybe I will ask this question, and you will take the next one, Eric. Yes, as you know, in Uganda, we did a lot of work until 2021, and we had identified VIC 172 as being a lead biotech potato, and we had actually already conducted regulatory trials. Unfortunately, the biosafety law, which we all expected that to be approved, has not been approved. And this is the reason why, at the moment, the situation in Uganda is kind of on hold until we have a legal framework that allows commercialization of GM crop. So for the time being in Uganda, we still need to wait until the law or a law is being approved. To you, Eric.
Thanks, Marc. There's another question that I'm going to handle. How have the growers responded to these cultivars? Acceptance, hesitance. We throughout the field trials in Uganda, also in Nigeria, we have had events where we invite growers to come and see how the biotech potato is performing in the field, and the acceptance or the perception is very positive from the growers, and some of the growers have sent their leaders who are positive about this, but the process has to go through the regulation before we can take it to the growers to try themselves in their fields. So it is very positive by the growers, and we hope we are soon going to have them growing this in one of the countries when it is approved.
And so let me take the next question. How is the local people opinion so far in GM potato? I think that this is a good question for you, Eric, since you've been participating to several events on this topic.
Yes, the local people opinion is highly influenced by opinion leaders. A lot of local people believe on the opinion leaders, what they tell them. But there are a lot of efforts around trying to educate people around the biotech potato and other biotech crops. So far, in countries like Kenya, there's been a lot of debates that we have participated, and we are continually educating them. A lot of the people have got a negative or misinformation and disinformation from various groups, various people, but we are trying as hard as possible to demystify this misinformation. And we hope that through our efforts, they are going to get the correct information about the biotech potato and other biotech crops that are being developed.
Okay, so I have another question. Did you try to remove the NPT2 gene from the triage stack? Are you planning on it? So let me answer this question, and Eric, you can take the next one.
Yeah.
We do not think there is an issue with the NPT2 gene because there is more than 50 GM crops that have been authorized for commercialization, cultivation, and consumption around the world since now 25 years that this technology is there. So we don't believe there is an issue. Of course, some concern have been expressed over time because NPT2, the NPT2 protein confer resistance to antibiotics, but it has nothing to do with what this gene is doing in the plant. And so we don't think there is a need for removing the NPT2 gene. Plus, if we set the precedent for something that scientifically is not really justified. I hope I answered the question. Up to you, Eric, the next one.
Yes, so the next question is, do you plan on looking at the durability of resistance with 1, 2, 3 resistant genes in a quantitative manner? So when we started the project, we started with single genes and we looked at each of these single genes separately and as a stack, 3 of them. And when we did the bioassay, we could see that the stack of 3 genes was giving higher resistance compared to when we had the single genes by themselves. That is in the lab. How they are going to happen in the field, we don't know if a single gene can give durable resistance or 2 genes or 3 genes. But the 3 gene stack that are in this biotech potato guarantee us that the resistance is going to be durable, because if a Phytophthora is going to overcome one resistant gene, then we have a backup of another. And if it overcomes, say, 2 genes, then we have another backup. So we have tested it in the lab, and we saw that having 1 gene as opposed to having 3 genes, the 3 gene stack was gave much better results.
Would you like to take the next question, Eric? Are you considering other countries in Africa who may be open to the biotech potato?
Yes, we are considering many other countries. Like you saw in our presentation, we have worked in Uganda, Kenya, and Nigeria, and we are looking forward to working in Ethiopia. And we have had requests from Rwanda. Actually, they approved this project, and we are looking for resources to work in Rwanda. And we are looking for other countries where the regulatory environment is conducive for this kind of technology to be undertaken. So we are looking in more than the countries that we presented.
So the next question is, nature abhors a vacuum. Do you see any other plant disease pests show up in GM potatoes? Well, the short answer is no, we haven't seen. We perhaps haven't looked hard at this aspect. But the fungicide that are being used are quite specific to Phytophthora infestans. So I do not foresee that there will be necessary any other disease that is going to come over that would be controlled by this fungicide. But this is something that we didn't look yet very much. Our observations are so far that no, there is no other plant disease that is gaining in importance. Next question for you, Eric.
Yes, the next question is asking if we are looking into other gene constructs. And yes, in the current project, we are looking into a new, or rather second generation construct with new sets of genes, and also including new new traits like PVY resistance. So in our second generation construct, we have PVY resistant in it. And also we are looking into other constructs that are going to have resistant to like bacterial and other diseases. So our focus is going to be not lead blight only, but also bacterial wilt and PVY resistance.
So there is one technical question about which one are the three genes. So we said VNT1, BLB2, and RB. So there are two genes from Solanum bulbocastanum and one from Solanum venturi. Let me take the next question. About a technical aspect about which modification was being used. It's not, we didn't use gene addition, it's the genetic engineering. So it's transferring the stack of 3R genes using Agrobacterium to Mephaceae. So it's, let's say, the classic GM technology. By genome editing, so far we cannot engineer, we cannot obtain complete and durable resistance to lead blight. This remains to be demonstrated. So there's still research ongoing before we can switch to this technology, which might be simpler to obtain acceptance. To you, Eric.
Yes, the next question is what is the difference in yield, users and consumer taste of the biotech potato compared to existing varieties, particularly in Kenya? So in the presentation, we saw that without use of fungicides, there is dramatic increase in yield of the biotech potato. And also one of the studies that we have done and we will do for the other lead biotech potatoes is that we're going to do the composition analysis. And the composition analysis basically looks at all the parameters that are in the nutritional parameters, which leads to the taste of the potato, and compare if there are any changes of the biotech potato and the conventional variety. If there are no changes, then the taste of the potato is going to be the same as the conventional variety.
Okay, the next question is, will the price charged to get access to these GMO seeds be significantly higher? Of course, we are not yet at the stage of the commercialization, so a specific and precise answer is not possible. However, the technology is public domain technology. It's not being, we do not need any return on investment on this technology, and therefore there is no plan for requesting any royalty on the technology. So most likely the price of the seed will be the same as the conventional seed. That's at the moment what we think is going to happen. To you, Eric?
Due to loss of chemical actives for lead blight control, as well as reduced sensitivity to other actives in Uganda, in Europe in particular, does it look possible that this form of technology will eventually have to be accepted there? I think this is for you, Marc.
Yes.
Acceptability in Europe.
Yes, there is in Europe a strong movement to ban all pesticides in agriculture. So there is a push for that. Of course, it has to be done on a case by case, and wherever it's possible, it should be applied. And I think this is a case where, yes, the answer is yes, it is possible to get rid of fungicide for controlling lead blight in potato. Now, will Europe eventually adopt? Nothing is clear on that side. We can see some clear interest in the UK to this technology. UK is not anymore Europe. That's one of the aspects that may make the agriculture going in its own way in the UK. On continental Europe, I think it will depend on those organizations that promote the end of pesticide use, whether they revisit their position on genetic engineering. I haven't seen a real push in that direction. There are, of course, some debates and some shy move in that direction, but I don't think this is going to happen anytime soon. But there is hope, at least I would have hoped that one day this kind of technology will be accepted in Europe because it contributes to reduction of pesticide uses. For you, Eric?
Thank you. Next question is, are there known genes for complete resistance to bacterial wilt? Yes, Jonathan Jones of the Sainsbury Lab in the UK has two genes, GM1 and ROC gene, that are showing complete resistance to bacterial wilt. And in this project, we are going to test these genes and see if they are going to provide complete resistance to bacterial wilt in the lab and also in the field, lab, greenhouse, and field.
So the next question is, do you have access to more R genes than the ones you have already introduced in VIC 172? In case the durability of resistance isn't as great as you anticipate. The reason I ask this, if my memory serves me well, I saw tuber blight in Toluca in Wales around 2009 and 2010. And I think you said Toluca carried one of the genes of Victoria 172. It's a very interesting, very useful comment because it shows the main issue with conventional breeding that usually focus on one R gene, not always only on one, but most often on one R gene. In the case of Toluca and Bionica, it was the RPI-BLB2 gene that is present, that has been shown. And indeed, there has been some report that Phytophthora, there are some strains of Phytophthora that are able to overcome the resistance in those varieties. So I heard that as well. In the case of our biotech potato, we have 3 R genes. So we think that it's going to delay the time by which there will be virulence developed. We do not know if that virulence will be as damaging as Phytophthora does nowadays. It might become a mild strain, a low aggressivity strain, which might not require to change the variety. But if we see that there is an economic damage, we will shift to other genes. And yes, the answer is yes, there are other genes available. We have, in the second generation products, we have a set of new R genes, those from Solanum americanum, isolated by the Sainsbury Lab and Jonathan Jones leadership. So yes, we have access to some of those genes, not all of them, but some of those we have access. Next one for you, Eric.
Next question is, what are the costs of producing LBR potato? Are there any other companies producing similar technology? Let me answer the second part of the question, then Marc, you'll do the first part. So we have in the US Simplot that has commercialized the innate potato that has some of the genes. So it's a similar technology, and this potato has been approved. The costs, maybe Marc, you can answer that.
Yes, sure. We actually produced a paper a couple of years ago testing, trying to estimate the cost of producing and of deregulating one potato variety in one country. Because at least at that time, there was the feeling in many of the environment where we operate that GM crop is too expensive and should be only developed by the private sector. the multinational corporation which have enough resource to do so. And so we did come up with the total cost for producing the potato as well as deregulating the potato of 1.5 to 1.8 million over a time period between 6 and 9 years. So that's why in my last slide I was saying it's within reason. And this is particular to the case of late blight potato, which is a crop that is mostly locally consumed. There's not so much export of the potato. There is export, but not the same as, for example, production of maize. And therefore, you can perfectly deploy in one country without having to obtain regulatory approval for all the neighboring countries. Also, it's one event, and it's as I mentioned, the risk assessment studies that are needed are relatively few and within the reach of institutions like us. We have done all the molecular characterization by ourselves. We are doing the composition analysis locally, and of course the agronomic and environmental impact are done locally as well. So that's basically the conclusion of that was my second point in one of my last slides, that this is within reason.
The next question is, according to you, will the industry be ready to accept the transformed varieties? The varieties that we have transformed or improved, these are farmer preferred varieties. They are not for processing. We have not transformed processing varieties because the industry, most of the potato that is consumed in these countries are table potato, and we haven't gone to varieties that are used by the industry for processing, for chips and other products. But we are looking forward to, if there's need, we can easily transform them and they'll be ready for the industry also.
Yes, on that last question, I think the priority is to release stable variety, because that's the biggest, let's say, demand, and also to stay away as a first product from potato varieties that are preferred by the processing industry, so that we do not create an issue for this industry. It's not really clear whether the industry would accept, but maybe after good acceptance from the fresh market, it might be better at that stage to start to transform processing varieties. And it will have to be based on the demand from the industry, of course. So we have a few more minutes, so feel free to ask more questions in the next 1 or 2 minutes. We will wait a little bit. And if not, Eric, feel free to close the meeting. If seems that there are no more burning questions. So I think, Eric, we can end here this. Oh.
Yeah. Oh, there's a last one. Okay. How is the practice distinguish phenotypically a seed production a wild from a transformed one? So
how can you distinguish the GM potato from its non-GM?
Actually, you cannot distinguish them by looking with their bare eyes. It's not possible to distinguish the transgenic or the biotech potato and the conventional one. But we are developing tools that can be easily used in the field to detect these biotech potatoes. So we have immunostrips. We have developed on-field field detection tool for each of the events. So it will be very easy to trace these biotech potatoes once they are released. It will be very easy for us to identify that this is this event that or this particular biotech potato once they are released. So we are developing those tools as part of the for the commercialization of this biotech potato.
On this point, we think that the after the deployment, there is, because it has immunity to lead blight, there will be a selection of the biotech seed against the non-biotech, because farmers are going to grow the biotech potato without using fungicide. So any, let's say, mix up with conventional tuber will be eliminated by lead blight automatically. So it's a natural purity system that will favor the maintaining the quality and purity of the biotech potato. That's one big advantage in this case. So maybe we can stop here, Eric, if you agree.
Yes, thank you all for listening to this webinar, and we look forward to the next webinar in the coming days. Thank you very much for listening.
Thank you very much for listening to us, and you can always reach us by email, of course. We'll be happy to answer any question we didn't answer properly. Goodbye.